let's just get straight into it. Andrew, thanks for coming and joining us. Could you describe what you actually are in this space, just rather than me do it? You tell us, what do you like to be known as?
Okay, so ko Andrew Veale toko ingoa. I'm a wildlife ecologist at Manaki Whenua Landcare Research. Landcare Research is a Crown Research Institute, and effectively we do science for conservation in New Zealand in terms of terrestrial conservation primarily. And I particularly study mustelids and genetics, and so the topics that we're going to be talking about today.
And I asked you before we started this, why would you want to do this in New Zealand and when you could do it anywhere in the world? What's so wonderful about doing this kind of work in this country?
Well, the difference of doing conservation in New Zealand is that you actually can make a difference. Now, in most of the world, people have been there for a very long time, and so they've killed everything. And unfortunately, New Zealand is still declining. We are still losing species. And therefore, we can make a difference now in this research by actually changing that and saving things that are currently decreasing.
Thank you, Dr. Andrew. Away you go.
All right, so good afternoon, everyone. I hope that you enjoy this and hope that I say something interesting and useful. There is a little bit of noise around me, so I apologize for that if that gets in the way. So anyway, my name's Dr. Andrew Veale. As I said, I'm a wildlife ecologist at Manaki Whenua Landcare Research, and I'm going to be talking about genomics, stoats, and predatory New Zealand. So some of this will be my research, some of it will be stuff that's ongoing. I'll mention some other people's research, and then I'll give some opinions on a lot of these topics. I'll try and make it clear when I'm giving opinions, because I don't want everyone to go, oh, but Andrew Veale said that. Some of it will be opinion, some of it will be fact, and hopefully some of it will be useful. Okay, I will just go back. So some deep history for me. I did my PhD in stoat ecology and genetics handing in 2013. So I've now been doing stoats for a very long time. After that, I did some salmon genomics in Canada and some stonefly genomics in Otago, and that was learning a lot of skills. And then I worked at Unitec for a bit, and now I'm at Manaki Whenua. And so, yeah, I've been in stoats for a while. And just going over some of the stuff that I did in my PhD, what I did, it was genetics and modeling. There are a number of eradication campaigns that were on at the time, particularly on islands. So Secretarian Resolution Islands in Fiordland and Rangitoto Motutapu. And they wanted to know, were stoats swimming back? And so I did some genetics with something called microsatellites. And I was able to show that a stoat that arrived on Rangitoto a year after the eradication had come from the Auckland mainland. And there have been multiple ones since then. I also just did some modeling, which is this picture up to the upper right. And this is what predicts stoats on islands. And basically it's distance offshore and island size. And I was saying any islands that are within 3 kilometers have a reasonable chance of having stoats there. And it seems to just be that they swim. And a lot of people were quite skeptical because, statistical models and genetics, it's a bit opaque. And people thought, oh, they can't do that. It's a tiny little animal, a 300-gram animal swimming across to Rangitoto 3 kilometers. That's crazy. And so I collaborated with Professor Kim King at Waikato, and we swam some stoats. And when you watch them swim for hours, multiple kilometers, you start to realize that, yes, they really can swim. And this is on Secretary Island, some of the work that I did there. And each color represents a family. And this was after the initial eradication campaign. And the stars are the mothers, and then the circles are the kids. And so this is using relatedness to try and figure out, well, who's surviving, how many are coming across, And the pictures here, so there's a little litter of stoats, very cute. And then the one below that is actually a tooth. So if you take the tooth out, the root of it has these little rings, and you can tell how old they are. And that way you can get really fine-scale relatedness. But this is using microsatellites, which is a very old technology. And I was able to get some relatively good relatedness values, but it was tricky because often they're inbred populations, and I didn't have that many markers. So moving on, since I've been at Manaki Whenua, I've been leading the Stoat Genome assembly. So a genome is all of the DNA sequenced and assembled in nice chromosomes. So you can see on the left there, that is a tattoo I have of all stoats entwined like DNA going up my arm. It's here. I don't know if you can see it on me. Anyway, and then the central one is a carrier type of stoat. So stoats have 21 autosomal chromosomes plus the sex chromosomes. They're labeled here funny because this was comparing it to weasels, and they've split to, so 4P and 4Q, doesn't matter. And then this is actually the animal that we used for the genome. This was an animal that was live trapped in south of Coromandel, so south of Thames. And since it was alive, it was humanely euthanized, but then immediately it was flash frozen in liquid nitrogen, so we had the perfect sample. And then in research that was funded by the Biological Heritage Science Challenge and Predator Free New Zealand, we threw all the sequencing at it. Now, this is more complex than most people here would be interested in, but basically we used 4 different sequencing technologies to basically create a perfect telomere to telomere chromosome, perfect genome. And this is better than any genome that had ever been created 5 years ago, because effectively we had the perfect sample and huge amounts of sequencing. And this is all publicly available. If you want to go on something called GenBank, you can search through and look at it. There's lots of A's, T's, G's, and C's, and all of it's annotated. And this is now going to be used for a lot of research. Then how do we use this? What I've been doing is something called GBS, which is genotyping by sequencing. And what we generate is something called a SNP. Now, a SNP is a single nucleotide polymorphism, which sounds complex, but it's just a variable site. So some individuals will have a C. So we've got A's, T's, G's, and C's making up DNA. Some might have a C, and some might have a T at that location. So it's just variation. And what we do with GBS is we cut up the genome with a restriction enzyme that chops it at a specific sequence. And so now it's in lots of pieces. And then we sequence the bit next to it, next to the cut, and look for these variable sites. And of course, we can map those sites back to the genome, so we know exactly where they are. And this has basically created a quantum leap in genetic resolution. Now, during my PhD, which wasn't that long ago, I'm not that old, I used 17 variable markers, 17 microsatellites, which is slightly different, but we can just, it's basically 17. And so for Waiheke Island, after some very stringent filtering, I have 30,000. And for Taranaki, I have over 55,000. So you can see that's a lot more. And what that means is that we can actually do a lot finer scale relatedness. It's not, are these two kind of related? It's like, what is the specific relationship between these two stoats? And that becomes very useful and important. Now, with Waiheke Island, I actually included that in some of my PhD research, and you couldn't tell anything apart because Waiheke Island is extremely inbred in terms of stoat populations. And so they all look the same if we just had 17 markers. Using 30, you can get a lot more information. Now, one of the things that I've done is just stoats across the country. And interestingly enough, we were able to get resolution so that one's in a PCA, you can get latitude versus one of the principal coordinates, which means that if you gave me a stoat, I would be able to tell you what latitude it came from in New Zealand with a reasonable accuracy, which is really exciting and interesting. And that might be because there are genes that are selected differently in the two islands, such as ones for going white in winter, because you definitely don't need to do that in Northland. It might be, coat thickness. It might be the time that they breed. Or it could just be random things that it's just isolation by distance. Anyway, we've actually started to find these things, which is very cool and interesting, something that we could never do with the old technology. Something else that we can do, of course, is genetically sex all of the individuals. One of the ways that we can do this is looking at the number of reads. So we sequence, millions of reads per individual, and then we can look at the depth of those reads, how many in each location. And of course, males only have one X chromosome, females have two. So if we look at the ratio of the reads on the X chromosome compared to the autosomes, it should be about 1 for females, and it will be less than 1 for males. It's not actually 0.5, and there's technical reasons why, but you can tell males from females really well using this method for sex ID. Now, you can sex stoats and weasels and everything quite easily if they're fresh, but if they're a pile of black goo or just some bones and hair, it gets a little bit harder. You can use genetics. Now, another thing that, of course, you can do is just go, well, how many reads do you have on the Y chromosome? And this works very well. You can see there's basically none for females and there's some for males. But I'm also doing weasels using the Stoat genome as a scaffold for this. And there was one female, in terms of having 2 X chromosomes, that also seem to have a Y chromosome. And that's Kleinfeldts Syndrome, which is a known genetic abnormality. It happens in humans. They often have intersex features. And I don't think that any mustelid has ever been recorded with Kleinfeldts Syndrome. I don't know anything about this individual. It was just cool that we can start to see weird things like this happening in a genome. So, now looking at relatedness of stoats. This is a heat map doing all versus all comparisons, looking at relatedness. The closer to red they are, the more related. And so on the diagonal, that's self versus self, and of course that should be highly related. The blue boxes, I've put in those to show you things. Now down at the bottom left, those are all the Waiheke stoats. Then there's a couple from Europe, which we'll ignore, and then there's some from Auckland and some from Taranaki. You can sort of see that there's little square structures inside that. So if we look at the Taranaki ones, those are clusters of highly related individuals, which are probably litters. Now, the big thing that you'll notice from this is all the ones on Waiheke are incredibly highly related. If you were just to go, are these two related or not to each other? You'd say they're at least first cousins, if not siblings. Every stoat on Waiheke is that related. That's because Waiheke Island has very little gene flow from the mainland, which means that if we remove them, they're probably not coming back anytime soon, which is wonderful. Waiheke Island's about 5.2 kilometers off the coast. There are some stepping stone islands which currently don't have stoats on them. And, I mean, results like this, it could have been founded, 70 years ago by a pregnant female. They are all clones, which is what I found with the microsatellites way back in my PhD. This is some information. So this is using ones from the Auckland region. So the Waitakere ones are from Ark in the Park. The newer ones are largely from the sort of Kokako management area. And there were some that I got from my PhD that were pre-eradication rangatoto. And then there was one that was caught back in the day in 2010. And that looks like it's sort of similar to one from Long Bay, which kind of makes sense. And then there were several ones that were caught recently. I've only genetically analyzed one of those, but it clearly came from East Auckland. That doesn't mean it came from the Hanuas, but it came from populations linked to that. and probably came up the Tamaki River, across the Browns Island and then maybe across. So you can start to go, well, where did a stoat come from using this genetics? And that was some nice results so that we know exactly where we should protect. So probably we should be trapping along the Tamaki River, maybe Browns Island as well. So here's some results from Waiheke Island, and I've been doing a really big deep dive because they are trying to eradicate there. They're having some great success, and they have been very kind and given me every single dead stoat, and so I am doing genetics on all of them to try and figure out what's happening. First off, these are the weights of them, and you can see that males are much bigger than females. There were 2 very, very little males, and they were caught when they were very young and probably still with mum. But it's almost complete, you can just say by weight, is it a male or a female for most of the year. And that's nice. It's just we're getting so many stoats, we start to get really good stats. Now, this is using those 30,000 markers that are selected to be really informative on Waiheke Island. And on this left graph, we have genomic distance, which is how related they are, we can say. So anything over about 0.3 is probably at least siblings or parent-offspring pairs. So anything in this yellow box that I've put on. And you can see this is the distance in meters on the x-axis. And you can see there are ones that are closely related, almost as big as Waiheke Island itself. So they are going quite far. But there's some really cool stuff that you can dig into. If we look at the frequencies compared, with distance. Now you have lots of ones that are very close together. Often these are litters that haven't dispersed yet. So you're catching the litters when they're very young next to mum. But there's this second peak that's between 3,000 and 4,000 meters away, so 3 to 4 kilometers. And something that we've often thought with stoat dispersal is that what they do is, they hang around with mum and then she kicks them out of home and then they run away. And it looks like they generally, if there's not too many other stoats there, are running about 3 to 4K away and trying to set up there. During the running there, they're probably not going to go into traps. They're not looking at things, they're averse to novel things. But when they settle in a novel place, they'll start to investigate and then they'll go into traps. So this is a really cool thing that no one has ever really seen before. What's the sort of average distance that they're going? Now, this is in a highly trapped population with traps everywhere. And so most of the time, it may be that they're doing something slightly different in other populations where the stoat density is higher. But they're also going up to 12K away. So we're starting to get the dispersal distance and patterns and behavioral patterns using genomics. Now, this is Waiheke Island if we just look at it itself. Now, as I said, if we looked at this thinking it was relatedness on the mainland and putting them all together, you won't get anything. But if we just narrow the data set down and recalculate everything, you can start to get these cool patterns. So again, on the diagonal, that's self versus self, and these square structures, like the one I've highlighted with the blue square, are probably litters. So red is highly related, yellow is less related. And so you can see we're getting quite a lot of litters within this. And if we start mapping those out, I've done each litter in a different color. You can see these light blue ones. That litter has filled the island. But there are other litters like this yellow one that they're all being caught quite close together. So we can start to see how they're actually moving across that landscape and recolonizing it before they're trapped. Now, on Waiheke Island, there's about a 250 by 250 meter grid over the entire island. So for those blue ones to have dispersed like that, they have encountered traps and avoided them. Now, here is a particularly interesting little story which you can start to get where I get really excited about heat maps like this, and most people probably aren't that interested. But you can see some cool stories. So we zoom into this cluster. Now, there is one square of 4 individuals and another square of 5 individuals, and there is an individual that is shared between those. These are 2 litters with 1 individual shared between the 2. So there's 2 options. Either they have the same mother, or the daughter in one is the mother of the other. So let's have a look at the details of this. No, wrong way. Okay, here's the dates of capture for all of them. So the first ones, you see these 2 red ones down here in January 2020. So there were 2 caught there, and then nothing was caught for a while until August that year when a female was caught above them. So that's this red one sort of in the middle. Now, she was pregnant. So she was caught before she gave birth, and so that was nice. Then the next one that was caught was in November, this orange one here. 4 days later, a juvenile male was caught in the same trap. A couple of weeks later, one was caught nearby, and then a couple of weeks later, one was caught down in the middle of the island, and then eventually in March the next year, one was caught way over at the far western end of the island, which was 7.3 kilometers away. So what has happened is there was a litter that was born at the end of 2019. It was probably down sort of Whakanifa way, and they spread out and were caught, and then they slowly dispersed, and one of them stayed close. She then had a litter around the same area, and then you can see the others starting to disperse across the island. Now, this one that went across, that is heavily trapped area, and so they moved across a third of the island, avoiding traps. Really cool stuff, and no one's ever got any of this information before, because we're getting the information of how every single litter mate that is caught is actually moving across that. And so then we can say, oh, you haven't got every single one of them. Now, looking at Taranaki, Manaki Whenua are doing a project on dispersal of mustelids across Taranaki for predator-free Taranaki. Hopefully it's flickering a little bit for me. Is everyone good, Nick?
Yep. Yes,
okay,
fine.
Good. Excellent. So you can do it as I'm showing here with radio collars or GPS collars, or you can do it with genetics. Now, hopefully my screen stops flashing because it's getting pretty weird for me. These are stoats and weasels that I have genetic samples of from Taranaki that I have done genetics on. The stoats on this one are red and the weasels are orange. Now you can look at the sex ratio because I sex them all genetically, and for the stoats it's about the same, and for weasels there's a lot less females. Now female weasels are about 50 to 60 grams, and DOC 200s you set them for about 100 grams, and this is highly statistically significantly different, and that means that the current trapping is not catching female weasels largely, and that's something that we all knew would happen. It's very, very hard to trap female weasels because you have to have the trap so sensitive they'll go off with any movement or with mice, and then you don't have any traps open because they've all gone off because of mice. So that was an interesting finding. We knew it would probably happen. And here's one of these nice graphs again, and you can see again, if we look at the right graph, the highly related individuals that are within this yellow box, generally they're quite close together, but there are ones going out quite far, up to 40 kilometers away. And so this dispersal curve here, it's complex. They are trapping less intensively than Waiheke Island, and also have more samples from the sort of beginning, so you can get ones that are in, it's more of a steady state environment rather than an eradication environment like Waiheke Island. But what Tom Etherington and I are going to be doing with this is looking at least cost pathway modeling. What this is that you try and inform a landscape model with landscape features and try and create a least cost path between all the areas, and then you'll be able to go, okay, here's how stoats will move. So then we can simulate and go, well, if we don't have any stoats in this area, how will they go? And then you can put traps where they will go, and then you can stop them reinvading, and you can get them. The thing is that when people do this, you can see this map is of the entire eastern part of the USA. I'm trying to do this at a resolution that no one has ever actually done, which is slightly daunting, but we're going to get some good results from that, I hope. Now, what does this look like? And again, it's flashing a lot for me. Hopefully it's looking fine for everyone.
Yeah, it's good. It's fine at this end.
Okay, that's good. So this is a map of both weasels and stoats. This one, the stoats are green and the weasels are red, and the lines linking them are links between close kin. So that's full siblings or parent-offspring pairs. And you can see there's some big movements here. These are sort of 40K movements. There's only a few of the very big ones, but I think that these are real. You know, a stoat has been recorded going 65 kilometers once. So they are capable of moving very far. I've looked, and the ones that are very far are adult males. So I think that these are the males that are the fathers of the litters. And so then they've been doing big movements trying to find females during the mating season. That's my guess. It could also be that the males from a litter are dispersing further. So I need to work that out. And also weasels, they're going pretty far. There's some 25-kilometer weasel movements across Taranaki. Something else that we're using all this stuff for is forensics. Currently collaborating with a group at Ecology. And we're developing forensic markers so that we can, you know, if you have saliva or scat, we'll be able to identify an individual from that. Now, a recent work that Ecogene did and I helped with was at Shakespeare Regional Park. And here, what happened was there was a litter of stoats born inside the park. And they were trying to figure out, well, how many stoats do we have? Where are they? How do we target everything? And so they got scat and also the bodies. And this is just using the old microsatellites. We were able to tie scat to individuals. And it was very good work by Talia and Julia. And there were, in the end, there were two individuals that hadn't been caught yet. And we sort of figured out one was these purple scats and there were these orange scats. So they were sort of, those were the two home ranges. And then a stoat was killed. And I said, that's probably individual B. And it was individual B. And then they've done a lot of looking, and individual A either left the park or died. So this was using a lot of things. So dogs were vital. Thermal cameras were very important. And then the forensic genetics of the scat that the dog found enabled them to say, well, should we keep going or not? And the thing is, incursion responses are incredibly expensive. You know, so it's very good to be able to go, we believe that there is nothing there, or there is a male stoat and it lives here. That's what you can start to do with genetics. I currently have a Marsden-funded project, which is being co-funded with Predator Free 2050 on embryonic diapause. Now, stoat reproduction is very interesting. Stoats, shortly after they're born, all the female stoats are mated by males that are just running around trying to find litters. So this is a juvenile female stoat. Her name's Aria, and she was donated by a member of the public. This is, you know, tiny little baby stoat, doesn't have her eyes open. She's pregnant. I know because she gave birth to lots of babies last year. And this is the growth curve of baby stoats. She was quite tame, and therefore I was able to get all these measurements. Now, you don't actually want to be pregnant if you're a tiny little baby female stoat. So what they do is they mate when they're very young. The mother is also mated at the same time. And then the fertilized egg sits there as a blastocyst, which is a ball of cells. And then it's arrested. It just pauses for For Stoat's case, about 10 months, and it just does nothing. It has this conversation chemically with the uterine lining, saying, oh yeah, just pause for a bit, and it's like, okay, cool, and then it just stops. And then it reactivates because of photoperiod, and then they become pregnant, properly pregnant. And that means that they're not really pregnant during winter, which was hard on the body, but they are all born at about the same time when food is available. That's great. Now, there's many species that do this. Mice can do it. Mink do it. There's various animals, and also a lot of wallabies. So my project is looking at how it evolved and how we can potentially control it. Now, it may be that we can get something in over that 10-month period where they take it and either it implants early and the uterus is not ready for it, or they're rejected. If we can manipulate a diapaused egg or embryo, then potentially that's fertility control. And that might be a lot easier because it's not necessarily making the animal sick. They could eat a little bit, have a bit more later, that sort of thing. And if you stop stoats breeding, you don't have any more stoats. So this might be a way that we can use genomics and fertility control to actually help control or eradicate stoats and potentially wallabies from the country. Now, my colleague Erica, she is working with me on using genomes to create novel toxins or potential novel toxins. And what you do basically is, if you have a really nice high quality genome, you then go, well, here are the proteins, such as the ion channels or the receptors. And so then you have the proteins. Then you can go, well, is there a difference between those in different species? And then we can go, well, okay, this one might be druggable and it's different for different species. Therefore, we could design drugs which are species specific, which could then be toxins and would not have a broad problem. And the one that she's particularly looking at are G protein-coupled receptors because they are druggable. They're one of the main things that we actually make drugs for. They're physiologically important, they're well characterized, and they could have species-specific pharmacology. And so basically what we do is we go from genome to gene sequence to protein sequence to protein structure to is there a difference, and then you express those proteins, you see if they act differently to a ligand, and then potentially we can make something from that. So that's really blue sky science coming from genomes that may end up being very good because we may be able to create something that's more humane, more effective, with less problems for other species. Now, something that when most people think, oh, genetics and control of invasive species, a lot of what's discussed in the media is a gene drive. What a gene drive does is it biases inheritance so that instead of it being a 50-50 chance, it's a maybe 70 to 30% chance of inheritance or a 100% 0% chance. If we link that to something that changes sex, then you can have only male offspring, only male offspring, only male offspring. And you could literally start with one animal and the entire species goes extinct. It's a doomsday switch for a species, which many people are concerned with. I should point out, there is no gene drive research being done in New Zealand. I am not involved in gene drive research. There is research on the modeling of how might this work in a population. And there is social discussions of, well, is this something that we might want to pursue? It's a very complex social and ethical issue. But I just thought that we need to mention it. Now, life history really matters for gene drives. And the main thing is generation time. So for rodents like mice and shiprats, the generation time is in weeks. That's quite good because we're talking, say, 30 generations is what it requires for an effect to have on a population. Stoats, if we're talking about males, it's a minimum 2-year generation time because males aren't fertile and then they convert, and it's an annual breeding system. Ferrets are similar, and possums it's 2 years as well, and they've got a long life expectancy. So when people talk about gene drives as a solution to problems, you have to remember this. And this is some modeling that I've been doing with Thomas Prouse over in Australia. And for mice and rats, in 6 years or so, if you had a perfectly working gene drive, now remember, this is imaginary future technology, but if one existed and it worked perfectly, you could have population crashes within 6 years. That's extremely optimistic. The most realistic scenario for stoats is still over 60 years, and for possums it's probably over 100 years. They just live too long and they breed too slowly. And what that means is that if we were to rely on gene drives for stoats or possums as a solution, remembering that this all requires a lot of testing and to be developed, we're still talking about a century in the future. They will be here into the future longer than they have been here into the past. So gene drives are not a quick solution for them, but it's very interesting with mice and rats. And I hope that we do have those discussions about where this research may go. The thing is, it will be done in other countries first. Obviously, in Australia, they have native rodents and introduced rodents, and you don't want to kill the native ones, and it gets very complex, and the mouse plagues are really bad for people with money, and therefore it's more likely that it's going to happen in Australia before it happens anywhere else. I'm just going to briefly show you some of the stuff that I'm doing on possums, just because they're an ideal species for this. This is work I'm doing with Prairie Taranaki, and these are all samples that I currently have freezers full of possum heads. These are the samples that I have, and you can see they're widely scattered over kaitakis and then around major rivers. And we're trying to see, well, are rivers a good barrier? And how do they move across farmland compared to forest, compared to scrub, that sort of thing. So this is some work that I had previously done on some West Coast possums, and I can almost not see my screen. It's doing some horrible things, but hopefully you all can. Now,
yeah, we can.
This is a fast-flowing West Coast river on the South Island's West Coast, and the two sides of the river were completely separate genetically. This has gone many, many generations of basically no gene flow. Apart from two individuals that were on the wrong side of the river and a probable hybrid. And if you look at the one over here, the picture to the right, you'll see two blue arrows and a purple one, hopefully. They were caught right next to the bridge. So the only possums that ever cross this river cross it at the bridge. And very occasionally they do have offspring, which to me says, I mean, this is a river that you guys wouldn't want to cross either. But it means that we probably should be doing control on bridges, and we can use rivers as a boundary in the right circumstances. So it gets very exciting when you can start to do genomics and see these sorts of things. And this is another thing that I got from that data, which was dispersal in different environments. And on the farms, there's a lot of possum control. And what you actually see, this is genetic similarity versus genetic distance. And you see that the green line has a different shape to the red line and the blue line. And that's because possums either have larger home ranges or they're dispersing further in grasslands where there are very few possums because they can expand their ranges. Now, that's really cool behavior, and I'm doing that from dead possums and genetics. So it's really amazing what we can do in terms of this. Now, this is all opinion from now on. I think, I'm relatively well informed, but, it's still opinion. Stoats are the most charismatic species in the country. Look at the little face. Again, this is Aria. She's very cute. And, they are lovely animals that we shouldn't be harming, but they do need to not be here. So I'm not saying that we should keep them. We're doing some of the best landscape genetics ever on mustelids and probably any species. In terms of the resolution, we are going very fine scale to try and get this done. And forensics is very useful for eradication and the mop-up phase. Gene drives for stoats are not going to likely help us anytime soon, and so we need other things. I go to community meetings and they're like, don't worry, gene drives. It's like, we need to keep on focusing on other things and think about this. I believe that it's impossible to eradicate stoats with even tweaks to the current technology. We need miracle new technology because slightly better traps, slightly better toxins. The problem is they disperse up to 65 kilometers, they swim up to 5 kilometers. All the females are pregnant. Many of them avoid traps. And so it's not going to be a little changes will eventually get us there. We need big changes. Our best chances, reproductive control, that could be a really big thing. All you need to do is stop them from breeding for a few years and you won't have any more stoats because they live fast, die young. Toxic rodents, something that Zip has been working on, is a very useful thing. And then there's people working on sound and scent laws. So Cacophony is working on those. My colleague Patrick, Manaki Whenua, is working on them. These will help, but they'll probably help in mop-up phases. And places like Waiheke, hopefully they'll get the stoats off soon. And there will be the ones that will avoid everything, and we need to throw everything at those few individuals. And obviously dogs are wonderful because they're able to detect them when we can't. Stoats are ninjas, and I have literally been looking in a tissue box, and eventually it's like, oh, there is a stoat there, because that's how well they hide. They don't want anything to do with you. But we do need miracle new technology for stoats. Other mustelids in the country, everyone's always like, oh, we need to talk about stoats. We've only really looked at stoats in an eradication context, and we only really research stoats in terms of how do we eradicate them. There is control for ferrets, mainly for TB, but they're not trying to eradicate, they're just trying to get them down low for TB stuff. And we have good evidence that when we control stoats and rats, weasels take off. Weasels love mice. Mice are held down by rats, so once you remove rats from a system, mice go up, and weasels will go up, and stoats control weasels, so once you start removing the stoats, weasels go up, and there needs to be research on the interactions between these, and weasels just need so much more research. Ferret control needs proper research, and personally, I think that ferrets might be the easiest of the species that are targets to eradicate, because they're not on the west coast of South Island. They're not in the beech forests, and that's really hard country. So we've already got rid of them from the hardest bits of the country because they just don't like to live there. And they're kind of habitat specialists, and they kind of like to hang out where rabbits are. So maybe ferrets we can get rid of. But someone needs to be doing the research on it rather than just saying, oh, they're all kind of like we can extrapolate from stoats. This stuff needs research. And while I've been involved in a lot of this research, there's a lot of other people that have done this. Obviously, Prairie Waiheke and the Prairie Taranaki, they gave me a lot of the samples I've talked about. Ark in the Park gave me a lot of samples. Getting samples from all around the country for these projects. There's been a lot of funding from Prairie 2050, Biological Heritage Science Challenge. Worked with Ecogene on a lot of these projects, and a lot of my colleagues at Landcare. So I don't want to be like, I have done all the research on stoats. I am just the messenger, and there's a lot of work happening, and there's other people working on this and in other fields. So I'd like to thank you all for listening, and very interested in taking your questions.
What's the biggest stoat eradication to date in New Zealand?
The biggest successful stoat eradication was Rangitoto Motutapu, which is 3,800 hectares, but that was done entirely through Brodifacoum poisoning the rodents, and they all died. The largest successful one with trapping, its sort of Anchor and Coal Islands are about 1,000 hectares each. I think maybe Anchor's 1,500. So that's the largest successful. They have tried on sectarian resolution. That's 8,000 and 22,000 hectares, and they still haven't succeeded, and they are trying on Waiheke. I think that within the next couple of years, I think that there's going to be some ninja stoats that avoid, but I think Waiheke will eventually be successful. And so that will be the largest successful stoat eradication ever in the world.
Professor Kim King said,
Yes.
Marvellous stuff, Andrew. I'm so proud of you. I just had to put that in. Somebody else wants to know, do you still have, is it Aria, the little stoat?
So Aria is at the animal facility in Lincoln. She is a show pony. She likes to interact with people. She's very tame. She also is a vicious animal, and this is something that people need to understand. So literally, I have caught feral ferrets, male feral ferrets, and within a day, you can toenail clip them and they'll sleep on you. Ferrets tame, and they're tamer than kittens. Stoats, you can raise them from birth. Before they open their eyes, they think that you're their mother, and there will be screaming and blood. They never tame, but she's moderately tame, and she's still my friend.
Another one, do you still have use for mustelid samples to be sent to you?
I always, yes, I'm always interested in samples, but there's always freezer space issues. So if people have them, yes, I am very interested. The fresher, the better. My colleague Florian is looking at the Pheromones, so he wants really, really, really fresh ones. So if anyone's live trapping, get in touch. But yes, I am always interested, but that could be a dangerous thing for me to say.
Yeah, it could be. It could be. Here's one. Have you found certain traps are more effective than others? And if so, which ones? It's a big question, that.
I have done some modelling of the trapping on Project Yanzoon, which is over Abel Tasman, and we found that the double set Doc 200s were better than single sets, slightly, and there is evidence in terms of lures that say ferret bedding is a very good lure. In terms of traps, having a run-through style design is generally very good because they then don't feel so enclosed and they can see the other end. But the problem is that then you need to exclude animals. So having, I believe it's called a rotary run-through, is a good design, and they're trying to put sort of a cross of wires so that weka don't get in. So it's tricky because they don't want to go onto traps. They are very intelligent animals, and therefore you need to make it as open and friendly as possible while not killing everything else that wanders in.
Best bait, in your opinion?
Best bait? My opinion, so there's good evidence that ferret bedding is very good, and so this may be because they want to steal stuff from ferrets, and ferrets are stinky. Stoat bedding looks very good as well, and the way that the stoat on Motutapu was finally caught after a long time, it was using a combination of food lure, which was a penguin egg. We also had female stoat bedding and sound clips of baby stoats, which I recorded, and it was mating season, and it was a male, and finally, and also it was a very naturalistic setting, and all of that was attractive enough to finally get the stoat that avoided everything.
Well, Tim's written in best bait, fresh is best. That's what he's put.
Yes. So this is the thing is that, and I know from the stoats that I've dealt with, they will starve and be very angry at you if you don't give them the food that they want. And it's like, I've given you the food. And they're like, no, I don't like that anymore. So very fresh, yes.
Here's one from Dan. Least cost, it was during your presentation, he put least cost path suggests that the stoats have pre-existing knowledge of the whole landscape. How is that possible?
No, so the thing is that, yes, that there are issues with that, but there will be preferred habitat for dispersal. And so what we do is we model going, well, how have they moved? And then we extrapolate saying that that is the best habitat. And my guess is that things like hedgerows, riparian margins, that sort of thing. They don't want to go out in the middle of open grassland. They do want to go where they are hidden. And so the least cost paths will be, they will just move around in those areas, and that's the way that they will get to places. But we're doing it postscript, going, well, how are they likely to have moved, by correlating the genetic distance versus the landscape features.
Here's one. Is there an estimate on numbers of stoats and weasels in New Zealand? Is that just too hard to estimate?
There was, I think, an estimate from Kim's book a while back, and the thing is that it will vary hugely. So the numbers, in October, November are probably 4 times higher than they are in July, and that's just because they're all born and then they all die. And so it will vary a lot, and it will vary by habitat, and it will vary by the beach mast cycle, because during a beach mast you will have huge numbers of them born, and there will be lean years where none are born. So it's a highly variable number.
Cheryl says it has been said that mustelids are evolving to be larger. Is there any research being done on this?
I don't think that there's anything that has been done. New Zealand stoats in some areas, so in I think it's sort of the eastern South Island, are about the biggest in the world, and that's because the stoats in the UK are the biggest in Europe, and they were rabbit specialists, and we brought them from there to here. And so in areas where they eat mainly rabbits, they are as big as they get anywhere.
In your opinion, ground tunnel traps or tree traps?
I think probably ground. You don't want to be asking the animal to do much at all. I mean, for me, the best way to control stoats is by poisoning rats. And no one likes to hear that. They like to go, oh, we just want to trap things. But the thing is that if you trap, you are asking an animal to engage with something that's like an alien spaceship in their environment. And a lot of them will not want to do that. If you poison lots of rodents, then there's lots of rodents that run around that are their natural prey out in the open, and they will eat those because that's what they naturally do. So by far the best thing, and always, if you can get social license to poison rodents, that will be the best stoat control.
What's the cost of genome sequencing per stoat?
Well, we threw about $50,000 at the super high quality platinum genome that we did. But now to get a similar sequencing of a second one, it'll be $500. And for the GBS, which is what I'm doing to look at each individual and compare the relatedness, so that's the stuff I do on Waiheke and Taranaki, the sequencing costs are about $60 per sample. Now, obviously, there are other costs beyond that. But that's the thing. If you invest in getting a really high quality genome, then the cost of everything else comes down. You can start to get the numbers up.
Jane asks, is anyone researching the pheromone that attracts the males to the newly littered females? She says it strikes me that this lure would be very effective.
It probably is. And so Patrick has been doing some scent trials at Manaki Whenua, and I still have bags and bags of scent, which is mainly socks and other things that Aria decided to have.
Kevin asks, if Oz doesn't come up with some way to sterilise stoats, what time frame would New Zealand be looking at to develop something?
I mean, sterilising stoats, so Australia's not doing that. They probably, I'm predicting, will... There are international groups in both the US and in Australia working on gene drives for rodents, specifically for mice, and that will eventually move to rats. For stoats, I mean, we're looking at everything, but this is 20, 30 years away for stoats, I'm guessing, because the research program will be a very long time. So it's tricky.
Rosemary asks, what is the estimated average lifespan of stoats and weasels?
Again, it depends on what you count as lifespan. It's something like over 80% of them die within the first 4 months of life. They then have a mortality curve of about a quarter of them die each year. So, because I have about 6 kilograms of microscope slides of stoat teeth from Resolution Secretary Islands, I got 2 or 3 that got to 5 or 6 years old. It really is a live fast, die young lifestyle.
Is there potential for future stoat contraceptives or abortive compounds to be distributed in rat baits rather than stoat baits?
I mean, this is the thing, is how do you get it into them? And maybe there's also the possibility, I mean, if we go full sci-fi, you could genetically engineer them to carry it, or you could genetically engineer bacteria to be inside the rats, which then produce it. But that's all science fiction and definitely not what we're working on, so don't be scared, everyone. But yes, there are possibilities like that in the future.
If we've got any more questions, and many more questions keep coming, how do we get in contact with you so that we don't physically ring you up?
So you can have a look on, you can contact me via Twitter, so I am Dr. Stoat. And also my email was on the last slide, andrew.j.veale@gmail.com. So if anyone wants to talk stoats, I'm happy to talk stoats.
And I suppose finally, because you've done so well, you've been going for nearly an hour non-stop, it's fantastic. What do you say to people that are in this space, that they're looking for motivation to continue in this space? What do you say to all of these folk?
I mean, when I was doing my PhD down in Fiordland, I was going across the islands, and they don't have 0 stoats yet, but they have succeeded in getting them very low. And there was birds on the island, and there was silence on the mainland. And we were doing coastal traps and going between the two. And that was very early. So you can make a difference. And basically, we have no choice. We have to be controlling these things. And it does. I mean, go to Rangitoto Motutapu and see what has happened in the now 12 years since the eradication. It's amazing.
Yeah, and so are you. Thanks so much, Dr. Andrew Veale. You are a tremendous leading light in what you're doing. And wouldn't it be great to think that in New Zealand we developed these incredible new ways before anybody else?
Thank you.
Now we've got all these lovely hearts and thumbs that come up, the new part of the webinar. So they liked what they heard. Thanks very much. Keep up the good work. Thanks, everyone.
Thank you.
Get in contact with Andrew or us here at Predator Free New Zealand, the Predator Free New Zealand Trust, and we can get you in touch with anyone you need to know. That's what we do. In the meantime, go and have a lie down, Doctor, and we'll see everybody next webinar. Take care, everybody. Keep safe. See you next time.
Thank you.
Bye-bye.